everyone, this is Chen. So here's how the story begins. Just last week, my friends and I were hanging out, and one of my friends bragged that he didn't attend the class yesterday because he drank a cup of milk tea in the morning and then had diarrhea twice, and his whole body felt bad. So because he's lactose intolerant. My friends and I were shocked. So, how could he drink milk tea while being lactose intolerant? But the truth is, according to the 2016 dietary guidelines for Chinese people, 90% of Chinese are varying degree lactase deficient, and 30% are truly lactose intolerant. But that hasn't stopped the Chinese from drinking milk tea, with the sales reaching 30 billion cups a year in 2019 alone. So, what's an enzyme, and what does lactase do that makes milk so bad for those who like it? Enzymes are biological catalysts that speed up chemical reactions in our bodies. They are highly specific, meaning they only catalyze certain reactions. This specific comes in four types: absolute, group, linkage, and stereochemical. Enzymes can also be categorized into six principal categories: oxidoreductases, transferases, hydrolysis, lyases, isomerases, and ligases. Each enzyme has its own corresponding substrate. Each enzyme has an active site, a region where the substrate binds and the reaction occurs. The active site and substrate fit together like a lock and key, ensuring the enzyme only catalyzes the right reaction. Enzymes work by lowering the activation energy of a reaction, effectively speeding up the reaction rate. This process is known as catalysis. Compared to the process of the enzyme, although reaction can still be carried out, the enzyme can greatly accelerate the reaction. Now let's talk about disaccharides. Disaccharides are a type of sugar molecules that are difficult to break down due to their complex structure. This is where enzymes come in. They help break down this complex molecule into simpler ones that our body can use. One such enzyme is lactase. Lactase breaks down lactose, a disaccharide found in milk, into glucose and galactose. Without lactase, our bodies would struggle to digest lactose, leading to conditions like lactose intolerance. Lastly, it's important to know that the activity of enzymes is influenced by the pH and the temperature of their environment. Each enzyme has optimal pH and temperature at which it functions best. Deviations from these optimal conditions can lead to decreased enzyme activity or even denaturation. In conclusion, enzymes play a crucial role in our bodies, from breaking down complex molecules to speeding up chemical reactions. Now, we turn our attention to the experimental part. We will measure the catalytic efficiency of lactase at normal body temperature, 37 degrees Celsius, for different reaction times. In order to measure the value, we use a UV spectrophotometer, which detects the absorbance of a solution in a certain wavelength range and reflects the concentration of the solution by this absorbance. However, neither the reactant lactose nor the products galactose and glucose absorb light in the UV wide spectrum. Lactase as a catalyst does not change with the reaction either. To do that, either the substrate or the product of the reaction must absorb light in the UV wide spectrum. To solve this problem, we are going to use another molecule with beta-glucosidic linkage that mimics the shape of lactose and can be hydrolyzed by lactase, also neutrophenol beta-D galactopyronoside, or OMPG for short. We will measure how much also neutrophenol is produced in the enzymatic reaction in a fixed amount of time by measuring changes in absorbance. Now let's get to the actual experimental steps. First, just crush one lactase tablet containing 200 mg of enzyme to an even powder using a mortar and pestle. Second, place the resulting powder onto a diagonally folded weighing paper and transfer the powder into a 15 ml tube labeled as suspension. Third, add 10 ml of phosphate buffer saline into the tube to suspend the powder, and then vortex the tube for 1 minute to maximize enzyme extraction. And now label two 1.5 ml centrifuge tubes, lactate suspension, and transfer 1 ml of suspension into each. 6. Just centrifuge for 1 minute at 10,000 rpm to sediment solid particles. And pay attention, the tubes must be placed in sources exactly opposite each other for the centrifuge to run properly. And finally, transfer 500 microliter of the surfactant from each tube to a clean 1.5 ml tube labeled lactase extract. And now we have 1 milliliter of extract in one tube at the end of the procedure. After all, prepare 14 1.5 milliliter microcentrifuge tubes. Label it as described in the table here. And add PBS buffer, OMPG, and lactase extract in each tube as described in the table here. And tap the tube to mix it and immediately place it in the dry bath set at 37 degrees. After this, set your timer and incubate the tubes in the dry bath at 37 degrees for desired time and shown in the table. Then, when the time is up, immediately take the tubes out and add 500 microliter of stop solution to each tube to stop the reaction. Before we put the product solution into the UV spectrophotometer, we can clearly see the results of the experiment. The yellow color of the solution depends as the set time of the experiment increases. And now, when we put the solutions into the photometer, we can see that the absorbance of the solution increases linearly with the reaction time. So that's all. This is Jin Chen. Thanks for your watching.